Dear students, welcome to the EPG Pathshala. I am Dr. Rajkishore Sharma, an Associate Professor in Chemistry Department of Delhi University. Today, we are going to discuss about the module Analysis of Photoelectron Spectroscopic Data. This is under the paper of Surface Analytical Techniques 2. The topics that we are going to discuss today as follows. Number 1, we would be discussing the PES spectrum or the photoelectron spectra. Number 2, the splitting. Number 3, the limitations. 4 is the method to improve the resolution. 5th, information content in photoelectron spectroscopy. And the 6th, why stimulated spectrum. 7th is the stimulated spectrum of lithium, and 8th is the stimulated spectrum of oxygen. 9th is data analysis. 10th is binding energy of hydrogen molecule. 11th is photoelectron spectra of neon, xenon and hydrogen fluoride. At 12th point, we will be discussing the guidelines for the interpretation of spectra from free molecules, and third is the newer developments. Before the interpretation of data, we will study what is photoelectron spectrum. Photoelectron spectroscopy or PES spectra are plots of photoelectrons count versus binding energy, where binding energy usually has units of electron volts, kilojoules or megajoules or electron volt per mole. We should keep in mind that higher binding energies correspond with electrons in lower energy subcells, that is, electrons that are closer to the nucleus and therefore require more energy to remove them. A photoelectron spectrum will feature peaks at different binding energies. The area under each peak corresponds to the relative number of electrons that have particular binding energy. We will often assume all the peaks have same width and the height of a peak is proportional to number of electrons.
This talks about splitting, the one which results out of Russell-Saunders coupling. The Russell-Saunders term symbol is used to determine the differences in the initial and final states for the spectral transitions. Here, the types of splitting that one comes across are of two kinds. One, spin orbital coupling, and the other is multiplet splitting. Spin orbit splitting, when we talk about, it deals with initial state effect which occurs during photoionization when electron is removed from degenerate subshell. Never occurs for s orbital. PE spectrum represents spin orbit splitting for P, D and F orbitals as doublets for XPS. PE here, when I said, it stands for photoelectron spectrum. Then we come down to multiplet splitting. Multiplet splitting will occur during interaction between unpaired electron formed by photoelectron ejected out and an already pre-existing unpaired electron. Multiple states formed during this photoionization can be seen and checked.
One of the main limitations of PES instrument is the resolution. The problems in resolution comes from the four main areas. These are: number 1, the dimensions of analyzer; number 2, the width of the entrance and exit slits; number 3, other electronic and magnetic fields; and 4th is the contamination in the analyzer. Steps can be taken to improve the resolution, but there are some methods that sacrifice other factors or other parameters such as the sensitivity. Obtaining high spatial resolution and high energy resolution always comes at the expense of signal intensity. Methods to improve resolution are One resolution improving technique that messes with the sensitivity is changing the width of the entrance and or the exit slits. For example, in an SDA, these slits define the range of trajectories photoelectrons that enter or exit the analyzer. Decreasing the width will certainly cause the resolution to go up, but the smaller slit size will decrease the number of photoelectrons allowed in and out of the analyzer, therefore lowering the sensitivity. Another technique is the addition of electron optics in the instrument. So this comes as an attachment. The third method of improving resolution is specific to XPS and is the addition of an X-ray monochromator to the system. These monochromators eliminate satellite radiation from X-rays and are capable of narrowing the X-ray line width from about 1 electron volt to 0.2 electron volt. The use of monochromatic X-rays is to simplify the spectrum.
Then we come to know about information contents of PES, which we get from photoelectron spectroscopy. The information that one gets from photoelectron spectroscopy, or as it is called PES, is the most important information which we get from PES is about the energy. Further, insight into the electronic structure of molecules give information about the ionization band shape and ionization intensity. With the help of photoelectron spectroscopy of ions, we get precise details about sp hybridization and direct spectroscopic signatures about the electronic shell closing of anions. Talks about why simulated spectrum. Where is the necessity to have simulated spectrum? Most photoelectron spectroscopy is a solid phase technique. Having so many neighboring atoms will cause interference. For example, an electron promoted out of a low-lying orbital will leave a vacancy which can be occupied by outer orbital with associated production of photon. If that photon is of high energy, it might eject a more easily ionized electron in the same atom or nearby atom. This results in production of background interference and even spurious peaks. As a result of this study, PES spectra derived from solids would be impossible to interpret. Talks about simulated PES spectrum of lithium. An example here has been taken, which is that of lithium, which has 2 electrons in first S sublevel and 1 electron in the second S sublevel. So ground state electronic configuration of lithium is 1s2 and 2s1, which is indicated in this slide. It talks about relative electrons on the y-axis and binding energy on the x-axis. 2 peaks, which represents here, they represent electrons in 2 different subshells. This is what one gets when we look at the representative spectrum here. We'll further go on to explain the energy contents in various sublevels. Peak with less binding energy, that is 0.52 mJ per mole, represents single electron, whereas peak with high binding energy, that is 6.26 mJ per mole, represents 2 electrons. Further, peak with highest binding energy corresponds to the electrons nearest to the nucleus. Here, peak with highest binding energy corresponds to two core electrons which occupy innermost subshell, that is 1s, and the peak with less binding energy corresponds to valence shell electrons, that is 2s. So according to the analysis, electronic configuration of lithium comes out to be 1s2 and 2s1. That is how we write 1s2 and 2s1. We'll further explain this. Peak having highest binding energy, that is 52.6 mJ per mole, represents two core electrons in 1s subshell. Peak with 3.04 mJ per mole binding energy represents two electrons in 2s subshell. Peak with lowest binding energy, that is 1.31 mJ per mole, represents four valence electrons in 2p subshell. So according to this analysis, electronic configuration of oxygen comes out to be 1s2, 2s2, then 2p4, which matches the actual configuration that one has.
Next is the data analysis. Most electron spectrometers use online computers for operating and data collection. It is also desirable to correct the data for background, distortion of collection efficiency as a function of electron energy, and contributions from satellite X-rays if monochromator is not employed. Finally, it is most valuable to deconvolute the spectral data. The major problems that beset the deconvolution are knowledge of the line shapes and the background that are often quite difficult to ascertain with precision. Nevertheless, deconvolution is a necessary element of data analysis, particularly in X-ray photoelectron spectroscopy. The DuPont curve resolution, which uses analog circuitry, has been used effectively. Its function is in evaluating a variety of parameters. Its weakness lies in the lack of an objective criteria for the goodness of the sample. Mathematical methods have been discussed for the deconvolution and something of the data, with particular concern for data encountered in X-ray photoelectron spectroscopy.
Talks about binding energy of hydrogen molecule. This has been illustrated in this slide and makes it very clear what way the energy changes when we move from one sublevel to another sublevel. The photoemission spectrum of hydrogen molecule has its vertical ionization energy at 15.8 electron volt. Further explains this phenomenon. One can rationalize this number in the following way. The vertical ionization potential, which we talk about, consists of two parts. First, the ionization potential of hydrogen, that is of the hydrogen atom, which is 13.595 electron volt, and number two, 1/2 of the binding energy of the hydrogen molecule, EBH2, which can be determined by the calorimetric method, is equivalent to, or it's given by, I vertical is equal to ionization potential of hydrogen plus 1/2 binding electron energy H2. The factor of 1/2 takes into account that two electrons take part in the bonding. The second term amounts to 2.23 electron volt. Thus, a total vertical ionization potential of 15.83 electron volt is predicted, which is in very good agreement with the measured value of 15.83 again, which is similar to the one which we had predicted. Photoelectron spectrum of neon. It explains what way the peaks will arise when we look at the spectrum of this. The photoelectron spectrum of neon consists of only one level, which is split by spin-orbit interaction, yielding a splitting of 0.1 electron volt, and is similar to the spectrum shown for argon, except for smaller spin orbital splitting. The line width is an indication of the resolution. Spectra like this one are often used to measure the resolution of a photoelectron spectrometer. The photoelectron spectrum of hydrogen fluoride. So here the heading given is photoelectron spectrum of HF. For the symmetry reasons, the axis of this molecule must lie along one p orbital. Thus, one has a singly degenerate sigma 2 orbital along the bonding axis and a double degenerate pi 4 orbital perpendicular to that axis. This has also been depicted here in the figure shown along. The same explanation is continued in this slide. The pi 4 orbital is non-bonding, thus showing weak vibrational structure, and is therefore identified with the photoemission signal at 16 electron volt. The sigma 2 orbital is bonding in this case and thus has a rich vibrational structure. It is identified as the transition which is observed at 20 electron volt. Goes on to elaborate the photoelectron spectrum of xenon. Xenon 5p3/2 spectrum at high resolution is measured with a path energy of 2 electron volt. This has been shown in the diagram alongside this writing in this slide. The measured width is 5.1 milli-electron volt, which can be taken as a good indication of the resolution of the system. Explains the guidelines for the interpretation of spectra for free molecule. Explains the guidelines which one shall use for interpreting the spectra of free molecules. Electronic structure, which when one calculates for molecules with the advanced computer code available today, molecular structure calculations have achieved a remarkable degree of accuracy, and it is certainly always a very good first approach to use a recent calculation in order to reach an understanding of an EDC. Vibrational structure analysis is the second part. Comparison with an isoelectronic series is the third part, then one that one could keep it in mind while talking about the spectra of various species.
Now about electronic structure calculation for molecules. Electronic structure calculation for molecules with the advanced computer codes is available today. The molecular structure calculations have achieved a remarkable degree of accuracy, and it is certainly always a very good first approach to use a recent calculation in order to reach an understanding of EDC. Now about the new developments. Beside the helium first and second line sources, other sources with small natural widths but still higher energies will be developed so that all molecular orbitals can eventually be studied with high resolution. The variety of available soft X-ray sources will also increase. Some with natural width of only a few tenths of electron volts. Monochromators will be coupled to higher energy X-ray sources, that is about 3 to 10 kilo electron volt. so that studies can be made at lower surface depths without undue loss in resolution. Synchrotron radiation should come into its own, and studies will be possible over a large continuous range of photon energy, that is about 10 to 10,000 electron volt with good resolution. Now we will discuss about the photoelectron spectroscopy of liquid samples. The surfaces of liquids with relatively high vapor pressure are not easily as accessible to investigations with the many experimental methods which have been developed in recent years for the study of solid surfaces. One prominent technique of this kind is the photoelectron spectroscopy for chemical analysis using X-ray photon source, for which Shigban was honored by the Nobel Prize in 1981. Besides the problem of large vapor pressure in liquids, which makes it impossible to obtain the required vacuum without the sample itself being consumed by evaporation, even the volatile liquids, it is difficult to maintain a clean surface. In photoemission studies, the liquid is picked up by a rotating device, often a sample disc, and a razor blade scraper is used to provide a continuously clean and freshly exposed surface in vacuum. Stainless steel needles with conical shaped trundles as well as moving weighted wires have also been used. All such experiments, liquid surfaces were limited to equilibrium vapor pressure not greater than about 0.1 torr. By restricting these experiments to very thin liquid films, the charging of the low conductance liquids by the electron emission was maintained by the relatively low resistance to a metallic substrate. For species with low degree of dissociation, this may cause problems for the interpretation because the influence of a conductor or liquid interface can penetrate a distance of several microns into the liquid by the electrochemical double layer. For the photoelectron energy analysis, a commercial hemispherical electron analyzer with a mean radius of 100 nanometer was modified to accommodate two differential pumping stages, both 240 liter per second turbomolecular pumps, one of the electron optics region and the other for the analyzer hemisphere chamber. During the experiment, the entire spectrometer ultra high vacuum chamber was maintained at a constant chamber wall temperature of about 150 degrees C to prevent both the adsorption of water and other residual gases, and also the buildup of surface charge on the surfaces of the electron optics and in the hemispherical analyzer. In addition to the MU metal shield, the entire apparatus is located in a 3 by 3 by 3 meter cube Helmholtz cage to further suppress the deflection of electrons by the Earth's magnetic field by an overall factor of 20. Additionally, the scattering chamber is initially clad with an internally heated magnetic shield of 150 micrometer thick cobalt magnetic AA foil to reduce stray magnetic field from the nearby laboratory equipment. The photoelectrons were detected by a standard signal channel, this copper beryllium multiplier. The extraction of information on the electronic density of states, we call DOS, density of states, is commonly achieved by a measurement of the kinetic energy distribution of the photoelectrons, which is taken as a direct measure of the density of states. Koopman's theorem attributes the measured mean kinetic energies E kinetic of the spectral feature to the corresponding vertical ionization energies IP in a single electron excitation model via Ip is equal to h nu minus E kinetic. Molecular ionization bands in the photoelectron spectra of condensed materials show energetic shifts of the vertical electronic work functions with respect to the related gas phase spectra. In liquid experiments, these shifts towards lower ionization potential reflects the bond solvation energy of the ions in a dielectric gas phase spectra. Thus, the ionization potential of liquids, Ip, are related to the corresponding gas phase ionization potentials, Ip gas, and it is represented by Ip is equal to Ip liquid is equal to Ip gas minus delta E G minus 1, where delta E G minus I is called the gas liquid shift. Note that in the literature, delta G minus I is often called the phase transition shift. Delta E G minus I can be simply estimated using a model consisting of a sphere with ion radius R0 and charge Z embedded in an infinitely extended continuum dielectric constant epsilon naught. The difference in the electric field energy in the liquid compared to the gas phase is given by delta E G minus I is equal to minus E naught square Z square upon 8 pi eta naught R naught 1 upon 1 minus 1 upon E, where I in the denominator in the brackets arises from E vacuum. Differences in delta E G minus I for different molecular orbitals together with careful investigations of the relative intensities of particular molecular bands can thus be used to estimate the conformational arrangement at the surface of the liquid,
which continues to explain the spectra of some other species. Study of electrons ejected at a few volts above threshold may provide photoelectron spectroscopy with substantial enhancement in sensitivity. Uses of monochromatized X-ray sources in the gas phase will enable one to measure the photoelectron spectra of core electron with a resolution of 0.2 electron volts, allowing the full usefulness of measuring suitable chemical shifts to be realized at last. Photoelectron spectra will routinely be accumulated as a function of photon energy and angular distribution. Detailed studies will be made on molecular beam of oriented molecules using polarized radiation. Methods will be worked out for determining the location beneath the surface of elements in an inhomogeneous solid. So these are the conditions which one has to keep it in mind while working out the exact positions of these peaks that shall arise in photoelectron spectroscopy.
Now, students, let's summarize the topics that we have learned today. The plots of photoelectron counts versus binding energy is known as the PES spectrum. In PES spectrum, binding energy is usually expressed in kilojoule, megajoule, or electron volts per mole. The Russell-Saunders term symbol is used to determine the differences in the initial and final stages of the spectral transitions. Now, about types of splittings, that is spin orbital splitting and multiple splitting. The first type, spin orbital splitting, is purely an initial state effect which occurs during photoionization if an electron is removed from a degenerated subcell. Another type of splitting is multiple splitting, which arises when there is an interaction between unpaired electrons formed by photoelectron ejected and already pre-existed unpaired electrons. steps can be taken to improve the resolution, but this in turn affects the sensitivity. High resolution always comes at the expense of signal intensity. One resolution improving technique that then messes with the sensitivity is changing the width of the entrance and or the exit slits. For example, in an SDA, these slits are what define the range of trajectories photoelectrons may have when entering or exiting the analyzer. Most PES is a solid phase technique having so many neighboring atoms cause interference. As a result, PES spectra derived from solids would be impossible to interpret. Thank you.